Okay, right. So in the previous video, I showed you a way of being able to sequence a much longer piece of DNA by cutting it into fragments, sequencing each of the fragments, and then we had this task of putting it back together. Well, we realized that if we just did this for one chromosome and just cut it up into fragments, it wouldn't work. So instead, what we decided to do was take multiple copies of that chromosome and basically cut them up in different ways using a partial restriction digest, which is basically just putting two lower concentration of restriction endonuclease onto the DNA so that it will only cut basically so that it will cut up only some of the restriction sites on the chromosome, and basically that will result in all the different chromosome 1s being cut up in different ways. And then basically what we can do is sequence each of those fragments, and you'll get fragments which overlap basically, and that allows you to gradually build up the sequence of the entire chromosome. Okay, so that was a clever way basically of being able to sequence much longer pieces of DNA by splitting them into shorter pieces. Okay, so, but what we now need to do, we've said, okay, if we can sequence these fragments, we're in business. What we now need to discuss is how did they sequence these fragments. So basically, with regards to the Human Genome Project, they've taken all 23 chromosomes, they have copied them, so they've got multiple copies of them, and they've now done this partial restriction digest to cut up all of these chromosomes in different ways. So they've got all of these fragments that they now need to sequence, and then they've got the massive job of sticking it all back together by finding overlapping strands, basically, and getting the sequence of all 23 chromosomes. So, how do they sequence all the fragments? Well, basically what they do is they put them firstly into bacteria. And this is where the concept of a genome library is going to finally come back in. So basically what they do is they take a vector called a plasmid. So this is a plasmid. And basically this is going to be a vector for our piece of one of our fragments. So we're going to put each fragment basically into a plasmid. So we will cut the plasmid with the same restriction endonuclease as we use to cut the DNA. So let's say we're using EcoR1. The plasmid may well have one of these recognition sites for EcoR1, a GAA-TTC recognition site. Okay, so when we cut it, what will happen is the plasmid will get the same overhangs as each of these fragments have. So then what we can do is we can put these fragments into the plasmid, like so. So in goes our fragment into the plasmid. So let's say this is the fragment in green. We'll have it in green. So here's the fragment gone into the plasmid. And then what we do is we get the plasmid to go into a bacterium. So we put this plasmid into a bacterium. Okay, so let's say this is a bacterial cell here. Okay, so it takes up this plasmid. Here's the, oh dear, what am I doing? Here's the plasmid here in the cytoplasm of the cell. Right, okay, the way in which you get the plasmid to go into the cell is it's a process known as transformation, bacterial transformation. But the way in which you do it is you put the bacterial cell in a very high concentration of calcium. And the reason this is effective is because if you remember the structure of the phospholipid bilayer, it looks like this. It has polar heads of the phospholipids facing the extracellular fluid and also the cytoplasm, and then the hydrophobic tails of the phospholipids face one another and form this hydrophobic core of the phospholipid bilayer. So this is the phospholipid bilayer, okay? And these polar heads of the phospholipids, they have a negative charge basically. So let me colour those in. So these are the polar heads of the phospholipids, and they have a negative charge. Now, DNA also has a negative charge because of the phosphate groups of the sugar phosphate backbone, which have a negative charge. So how are you going to get a piece of DNA to pass the phospholipid by there? Well, if you put it in a really high concentration of calcium, calcium ions have a divalent positive charge. So let's show them here. So what will happen is calcium will form electrostatic interactions with the negative charges of the phospholipid heads, and that will neutralize the negative charge of the phospholipid heads. Then what might happen is the plasmid might also come over here and interact with the calcium, like so. And basically what you then do is you heat the bacteria up, so-called heat shock. And what just sort of happens is the phospholipid bilayer sort of gets disturbed by the heat, because remember, when things are hotter, the molecules are moving around more. So the phospholipids are going to move around more, and what just manages to happen is the plasmid sort of manages to make its way through the phospholipid bilayer as you make the phospholipids jiggle about more. It's not completely understood the mechanism by which this works. What is known is that it does work. You put them in a very high calcium solution, a concentration of calcium, and you then heat them up, basically. And it's known as heat shock. Okay, and it makes the bacteria take up the plasmid, so it transforms the bacteria so that it now has our plasmid containing our fragment of DNA in it. Okay, now what we do is we get a bacterium for every single fragment of DNA you've made, basically. So you have a bacterium containing every single fragment that you've made. So you've chopped up multiple copies of every single one of the 23 chromosomes, and you've chopped them up all in different ways. You have a huge number of different fragments. You create a bacterium for every single one. And when you've got a bacterium for absolutely every single one, the collection of all of those bacteria is known as a genome library, because basically it stores the information of the entire genome. Every bacterium has an individual fragment, and that's called a genome library. So now, the next aim of the Human Genome Project is to take out every single bacterium one by one and sequence the fragment that it has within it, the portion of the genome it has within it. And what's brilliant about bacteria is that they will copy these plasmids. So they're going to copy them up, like so. So you'll end up with bacteria which are full with multiple copies of the plasmid, basically, with our fragment in. So you'll get these bacterium which are which have produced lots of these fragments that you're interested in. And now the next stage of the Human Genome Project is merely going to be to sequence that fragment.